Okay, this is a quick look at some introductory stuff about cells. Most of you probably know this or have heard a lot of this already, but we're going to cover some of the basics pretty quick. Okay, a lot of what we call cell biology is also known as cytology. Cyto is the root for cell, and you might hear the words cytoplasm or cytologist is someone who might do that kind of work. So cyto means cell, so you might hear words with that in it. Just looking at the scale here, most cells require microscopes to see, and those are basic light microscopes, which is one reason why they were discovered pretty long ago, because the microscope's been around for a couple hundred years. There are some smaller structures that require electron microscopes. The bad part about electron microscopes is that you can't look at living things. So whatever you're looking at kind of has to be frozen in place. So there are some large cells, like you might see frog eggs in a pond or whatever. You might see chicken eggs are even considered single cells at very early in their development. So bird eggs are cells as well at one point. Okay, some other tools we use for cytology are these three ideas here. We might fractionate the cells. That's where we'll put them in a blender and then centrifuge them in stages. So we'll look at that. Chromatography, that's where we separate things based on their solubility. So you've probably done chromatography in high school chemistry, where you separate inks into their different pigments and they'll travel different distances through the paper. Electrophoresis is using electricity to pull particles through a gel based on their size and polarity. So here's what the cell fracking looks like. You take the cell and you homogenize it. And what that basically means is you put it in a blender and break it into tiny pieces. So you could do that with, say, liver cells and make what's called a homogenate. Then you're going to centrifuge this multiple times. So you'll centrifuge it and you'll draw out the top portion, centrifuge it, draw out the top portion, and you'll keep doing that over and over and over again. You can see the force of the centrifuge right here is increasing. Okay, so we have 20,000 Gs, 100,000 Gs, and 150,000 Gs, and we also increase the length of time we do it. As we get to smaller and smaller particles, we require more force and more time to centrifuge. So we can use just basically force to pull things of different sizes out, but we keep doing it in different layers. So we start getting out nuclei and some of the pieces we broke off. Then we get out smaller organelles. So if you wanted to look at these things individually, this is how you could get to them, all the way down to ribosomes. All right, let's look at the other methods of separation. Chromatography. We talked briefly about that, but we're separating things by size or degree of how attracted they are to whatever we are using to separate these different fragments out. So that could be paper, there's gas chromatography, column chromatography, thin layer chromatography, where things are pooled into separate layers based on a factor, usually size or attraction. Electrophoresis, let me draw a little bit of how that works here. You would take a block of gel and you're going to create little what are called wells, which are basically just holes. So that hole goes all the way down into the gel, and you fill the well with whatever you want to separate. And so a lot of times it's DNA. Then you're going to hook this up to electricity with a positive and negative side, and hook it up to some sort of power supply, and you'll let it run for a little while. And substances will start traveling down what they call the lane, and they'll sort of separate out into what looks like bars. It almost looks like a barcode, and these will be different amounts of different shades depending on the amount that's there. And so you can compare two samples to see if they're the same. And this was sometimes used in what we call DNA fingerprinting. There are more complex ways to do it today, but it's still accurate and it still works really well. Okay. We're going to go ahead and stop this video and make this a short one so you can break this into the parts you need.